Some of the plots that we're going to show you through today are mostly our fungicide trial. What we're looking at there is timing of the application and to see if there's any difference in efficacy. So our timings are daytime, which was 12 to 1 p.m., nighttime, which is 12 to 1 in the morning, and then early morning, which was 4 to 5 o'clock in the morning. The reason behind this is we have done a night spraying herbicide trial for the last couple of years, testing the same kind of thing, looking at efficacy at the application timing, and there has been some results there that show the early morning application that a lot of people have been trying is actually the worst time to spray. The reason in this part of the country, why do you guys think that we do that? Why do you think guys are getting up at ridiculous hours? The wind, exactly. And I think our results are starting to show that even if you start later in the day, if it's not a windy day, then you're safe. But the wind also dies down after supper usually. So if you guys want to get in the field sort of after dinner, go relax for a bit when it's really hot and windy, and then after supper, go out. And with the advent of the GPS, auto steer, all those things, if you have your fields mapped, you can actually spray into the dark, and you're not going to see the same kind of drop in efficacy that you do in the early morning application. So anyways, we'll take you through those plots. There's peas, wheat, barley, and canola, and I challenge anybody to go through the canola that we have this year. It's huge and it's really, really thick, and I've tried to get in there to look for disease, and I found in half an hour, I found one plant with sclerotinia. So if anybody else can find one, I'll give you a prize. So I'll turn it over here to Ron, and he's going to kind of talk about scouting and that sort of stuff. And then after he's done that portion of the talk, we'll get on the trailer and we'll let you guys out into the field, and you can look at the plots and have a look for yourself what exactly is out there and practice your skills.
Great. Well, thanks, Jamie, and thanks to Farming Smarter for hosting us today for our fall disease crop walk. You might be wondering, what's the point of looking at diseases in the crop at this time of the year? Isn't the water already under the bridge? The opportunities to use, say, foliar sprays for leaf spots past us. Well, that's true, but the fall is a good time to take stock of the diseases that are in your crop, in anticipation that some of those are going to survive through the winter and could possibly pose problems next year. So we're really, at this time of year, getting a glimpse of what we might encounter next year if the residue-borne diseases all survive the winter and they're there waiting for us the following year. This is also the time of year when a lot of the diseases reach their peak in terms of symptomatology. This is when you'll see the most leaf area affected by leaf spots. The root rots will have reached their max, and with the hot weather that we're having now, any plants with compromised root systems are going to wilt and possibly die. It's also the time when we often see reproductive structures of, say, fungal pathogens. We may see a lot, old striped rust lesions or even young striped rust lesions on the plants. We may see diseases like the bunts and ergots and fusarium head blight in the cereal, so it's a good time to do that. It's also a good time for farmers to take stock of what has and hasn't worked in their crops. So for example, if you've tried a new sprayer, maybe a new nozzle array, water rates, new fungicides, insecticides, those sorts of things, now's the time to determine whether those have worked. You can either do that by assessing the levels of diseases or insects in the crop, or if you have yield monitoring equipment on your combines, you can assess the yield responses and quality responses later on from those treatments. So if half the field was done with fungicide A and half with fungicide B, you can make those comparisons. So this is actually a good time to be mindful of diseases and their impacts on the crops. I noticed the recent issue of the Canola Watch magazine that there's a there's an article there on harvest disease assessments and looking at, and they give again a lot of the reasons I just mentioned for taking the time to get off the swather. If you come to a patch, for example, where the plants are dying or not doing well, take time to get off the swath, or maybe mark it to come back later and actually dig up some roots and see, is that club root, is it foot rot, or what's the reason for that? Or it's root maggots, maybe it's an insect, that type of thing. So this is what we're going to talk about today as we go out in the field. So the first part of our tour this morning after this introductory session will be to go to tour some of the Farming Smarter plots where we'll see diseases of cereals, peas, and canola. We've also brought some plant specimens and material here of things that you won't see in the field today. And we're going to spend the last 3/4 of an hour or so back here at the tent talking about some new and unusual disease problems. Bacterial and fungal leaf stripes on wheat, a new disease on corn that we found called Goss's wilt, and some other things as well. So we brought some literature along here. Some is displayed and there's multiple copies. You can certainly take some of those with you to begin to build your library of information on diseases and disease management. So as we go along today, although Dr. Harding and I will be probably doing most of the talking, really we'd like to encourage you to offer your comments on what you're seeing in the field, share some of your observations of what seems to be on the upsurge or maybe on the decline, things that may have worked or not worked for you if you're a producer, or if you're a consulting agronomist, maybe if you can share some of the triumphs and failures that you've seen out there this year. That all helps us learn about diseases and disease management. Okay, any questions before we start, or? Okay, well, we're going to board the wagons, and
Before you do, make sure to grab some booties and put them on before you head on the trailer, please.
right, we're sort of standard biosecurity protocol here is we'll put the boots on. This is so we don't want unwanted things coming here, or more importantly, maybe things going from here back to your farm. So just slip a pair of boots on, and when you come back in after the tour, we can discard them here.
All right, so this is a trial that actually Ron and Mike came forward to us with. And does Kelly have this one as well? Kelly Turkington. And so what we're looking at here is we have 16 treatments, and prothioconazole, tebuconazole, and Serenade biofungicide. So there's a bunch of different combinations of each one. We sprayed a set of it at flag leaf timing to protect for leaf disease. We sprayed some only at head blight timing, which is during flowering to protect for head blight. And then we sprayed some at both, and then we have a check. So this was seeded May 10th. Our flag leaf application was July 3rd, and head blight application was July 15th. I've got a couple samples I gave to Mike already. Ron's got them actually. I managed to find one possible head blight. It doesn't look like we had a big infection here, and maybe a few other things.
What's in the guard rows here?
Well, the guard row seeded in between is winter wheat, so we haven't had a chance to mow it out, but there's definitely a pretty big leaf disease infection on the winter wheat as well.
Good.
Yeah.
Okay, thanks for that explanation. Yep. So we'll give you a chance to walk through the plots here and have a look for diseases. Now here's the ones I want you to key on. Anybody know what that is? That's ergot, Claviceps purpurea. Oh, sorry. Okay. Yeah, I'll get you to hold that up. So this is ergot, Claviceps purpurea. It's a fungal disease, and it infects the flower and the ergot body, which is the reproductive structure of the fungus, replaces the kernel. Ergot is a disease that kind of ebbs and flows. In years when we get wet weather, especially around flowering, it seems to favor infection by the fungus. It usually initially infects grasses and then moves from the grasses to wheat and other susceptible crops with insects and some rain splashing and that sort of thing. The black ergot body that you see here is what survives in the soil, and they'll last usually at least a year or two. They're not as long-lived as Sclerotinia sclerotia, but they do survive in the soil. They germinate in the spring, much like Sclerotinia does, and they form a structure that releases spores, and that's what infects the grasses. So with ergot, we're concerned for it for two reasons. One is obviously you're getting a loss of yield when the kernels are replaced like this, but the major issue is that these ergot bodies are actually poisonous if consumed by humans or animals. They cause a disease called ergotism, which is essentially results in restricted blood flow, especially to extremities like feet, ears, that sort of thing. And eventually you may get gangrene and die from it. So in cattle, with tails, ears falling off, and hoof rots and those sorts of things, usually the animals have to be put down. This is the ergotism in the Middle Ages. They called it St. Anthony's fire because your extremities burn, but they have a burning sensation. They grew a lot of rye in those days, and rye is very susceptible to ergot, probably the most susceptible crop. And they ground the ergot bodies and ate them along with the flour, and that disease caused a lot of misery and loss of life.
Would you have to eat much to get sick?
You probably wouldn't have to eat that much. No, I would imagine, a handful of ergot bodies would make you very sick. Yeah. There's a medicinal value in the ergotamine steroid, in that it used to be used a lot in surgery to restrict blood flow and they used to administer it prior to surgery, and it induces abortions and so on in cattle and humans if it's consumed. So it has a lot of nasty side effects. Now they synthesize the material artificially. They used to buy the ergot bodies and process them from seed plants. That's right. Okay, so that's that one. Now this is Fusarium head blight. Mike will just hold that up. You can see the Fusarium fungus has infected about midway down the head, which is usually where the first flowering starts, at the middle of the head, and it's killed the rachis, and everything above that point is died. With a hand lens, we may be able to see some of the Fusarium fungus there. If not, what you can do is take a head like this, put it on a moist paper towel, slip it in a plastic bag, and blow the bag up like a balloon to create a little humid chamber. And in 2 or 3 days, that's usually covered with salmon pink mycelia and spores of the Fusarium fungus, especially Fusarium graminearum, which is under Alberta's Ag Pest Act a declared pest. And we try to restrict the spread of the disease through the province. Unfortunately, here in the south, it's become endemic, especially in our irrigated cereals, but it's rarely found in central Alberta and the Peace region. We have some fact sheets and the Fusarium graminearum management plan back at the Quonset there if you want to pick up a copy of that.
Pretty good year for Fusarium.
Right, this head also has some gloom blotch. You can see some black discoloration on the side of the head there. And gloom blotch can be due either to bacteria, and we'll talk a bit about that, bacterial gloom blotch, back at the tent there, or the fungi that cause leaf spotting, Septoria and Staganospora, will cause gloom blotch. And gloom blotch is a descriptive term for a blackening, discoloration of the glooms that can actually grow through the glooms onto the seed and possibly cause some discoloration there. The guard rows have lots of leaf spotting. This is mostly tan spot. Because winter wheat planted in the spring doesn't go to flower, it remains vegetative. It's ideal for guard rows when you're growing spring cereals because it stays stunted. It's a nice separation between the plots as Mike has done here. But the winter wheat is very susceptible to the leaf spots and to the rusts, and some of the best disease symptoms in plots are often not within the plot but within the guard rows, especially if it's winter wheat. Also, the winter wheat will show up barley yellow dwarf and wheat streak mosaic and things like that too if it happens to be around. Okay, so we'll give you a chance to walk through the plots. What do we got there?
Is there a major yield impact with the leaf spot disease?
Sorry, say that. I'll let you repeat the question there, Jane.
I was just wondering what kind of yield impact you're going to see with the leaf diseases.
That's a very good question. It really depends how far up the plant the infection goes. If it stays on the lower leaves, it will probably have very little impact on yield. But once it gets up onto the flag leaf and the flag -1 leaf, you can see yield reductions of 10%, 20%, even more if it's severe. So that's when you approach levels of infection that high, that's where fungicides will usually pay the well-timed applications to protect the flag and the flag -1. Now there's additional impacts other than just yield that these leaf spots will cause. Tan spot, when it gets onto the kernels, especially durum wheat, causes a disease called red smudge. The kernels take on a red discoloration, so that represents a quality loss in addition to a yield loss. And the tan spot can also get up onto the kernels of other wheat as well. Black spot or black spot on the kernels, where the end of the kernel is black, is usually caused by alternaria or helminthosporium, and or black point, sometimes referred to as black point. That's also another grade reducing disease. I don't think we'll see any of that here. Usually you have to look at the thresh grain. So in wheat we have, I guess we use the term leaf lesion complex in wheat because there's usually not just one type of leaf spot there. For example, along with the tan spot, you'll often find Staganospora leaf spot, which is another fungal disease. The two are often very difficult to tell apart. Earlier in the season, there's another leaf disease that quite often you see on the lower leaves called spot blotch, and that's caused by the common root rot fungus. It has a leaf disease phase as well, seedling blight phase. Usually it fades out as the plants get bigger. The Staganospora and Septoria take over. And as I say, once they destroy the photosynthetic area on the flag and flag -1, then that's when you start to see the big yield reductions happen. And they will also get onto the stems here and cause lesions there, as well as the glumes, as I mentioned. So they're actually capable of attacking all of the above ground portions of the plant. And what you'll notice on these, that the disease gets progressively worse as you go down on the plant, and the older leaves get sacrificed early because of the leaf spots. Now the tan spot and Septoria to some extent are what are called polycyclic diseases, in that they'll form spores on the dying leaves that will reinfect healthy green tissue. So they may actually go through several disease cycles during the season. That's what makes them explosive diseases. And they also go to native grasses and cultivated grasses as well. Even the prairie grasses, you'll see tan spot lesions on prairie grasses once in a while.
This field was in the grass alfalfa.
A grass alfalfa mix, yeah. That's right, so quite possibly carried over. But the airborne spores of these leaf spot fungi can carry long distances, and you basically, in southern Alberta, we're blanketed with these spore showers during the growing season. And provided there's moisture on the leaf surface when they land, they will germinate and infect the leaves. And humid, yes, high humidity and the mist hanging in there. Okay. All right, let's maybe take about 5 minutes and let you have a look through this plot.
As many of you know, there were multiple hailstorms that passed through here, and so there's a significant amount of pocked damage on the canola stems and some of the branches due to the hail. And one of the things that happens often after a hail injury is alternaria black spot can set in. It's a kind of a, I mean, it is a bona fide pathogen of canola, but if it can find wounds, it sometimes can become more of a problem. So alternaria black spot is something that can become a real serious problem when there's the predisposing wounding factor that hailstorms will cause. And so on this branch here, you can probably see from where you're standing that there's some blackening around where the hail damage was on this main stem. In addition to that, on many of the pods, you see small black spots. And so that's the alternaria black spot organism. And this can be a relatively economically damaging condition when you get a lot of spots or alternaria colonization of the pods, especially when the damage is early enough that it will start to cause discoloration of the, grow through the pod wall and cause discoloration of the seed, and even start to kill the pod and the developing seed. So there's some nice examples in this field of canola here of alternaria. Jamie and I did our best to find some evidence of sclerotinia stem rot. We didn't in this little corner. If we scouted, we might, but it is still, at least in the plots that I've been in, a little bit early. Probably by next week, especially if the field is irrigated and stays wet, we'll start to see some real evidence and symptomology of sclerotinia stem rot. I know that in some commercial fields it is evident now, but in a lot of the research plots and fields where the disease pressure oftentimes isn't quite as high as it is in commercial fields, and we don't have the large expansive canopy, we don't often see it as much or as quickly in the plots. So unfortunately we don't have any sclerotinia here, but back in the tent we've got some sclerotinia symptoms on canola leaves to show you. So other diseases that you could scout for in canola at this time of the year, it's actually the best time, as Ron mentioned in the tent, to sort of characterize what the disease situation in a field is. And so you can look for alternaria black spot, you can look for black leg fungus, you can look for club root, and you can look for sclerotinia stem rot. And just really the best time to look for a lot of these is just ahead of the swather. Unfortunately, that's the most difficult time to walk through a canola field, but this is when you'll be able to see the symptoms and really sort of characterize what the disease situation in a field is. Does anybody know how you would scout for blackleg? If we were to walk out into this field, how would we look for blackleg?
Cut a stem open.
Cut a stem open, exactly right. So that's blackleg fungus can attack all the above ground parts of the stem, all the way from the cotyledon early on. You can see blackleg lesions at that stage. All the way through the season you can see lesions, blackleg lesions on the leaves and the stems. But really the way to confirm that it's blackleg, because there are other organisms that can cause lesions that have the diagnostic characteristics other than blackleg that are not pathogens, the way to really confirm the diagnosis is this time of the year to cut the stem right at the soil level and then look at the cross section of the stem to see if there's any black discoloration. If there's black discoloration in the stem, then that's there really isn't anything else that will cause that symptomology other than the blackleg fungus. If you see a brown discoloration, that's probably Rhizoctonia or some other soil-borne organism that's colonizing at the crown. And if you see red or pink discoloration in the stem, that's more likely Fusarium. So there are other things that will cause discoloration of the internal part of the stem that you can visualize at the crown. But if it's black, it's most likely blackleg. And so this would be the time now. The blackleg fungus lands, the spores will land on the canola leaves, and then they grow internally through the branches and down through the main stem and colonize right at the soil level, and they form a hard stem canker. And so sometimes you can actually see those basal stem cankers, and it's a very hard woody symptom. So if you poke it with your finger, it won't be soft and mushy like a sclerotinia stem rot lesion will be. It'll be hard and woody. And those stem rot lesions, those basal stem cankers caused by the blackleg fungus, if you cut those open, you'll see the black internal discoloration. And sometimes, depending on when you catch the infection in the life cycle of the pathogen, you can start to see the blackening of the stem before you see the basal stem canker. So that's why now is a good time, because even if it hasn't started forming basal stem cankers, you could still see the blackening of the internal vasculature or stem tissues. So if we were going to scout for sclerotinia, how would we do that? Better than Jamie and I did, because we couldn't find it. What would you look for, and where would you look?
Where the leaves attach to the stem.
Okay, so the comment was made here where the leaves attach to the stem. Yeah, that's a great place to look. Why? Because what gets lodged in there? The petals, the blossoms, the flowers. And that's really the food for sclerotinia to get started on. And you'll see that in the tent, the demonstration we have there with sclerotinia. You can see the old decaying blossoms that are stuck on the leaf surface, and then just chlorosis emanating out from that, where the sclerotinia fungus is starting to grow out into the tissue and kill it, but we don't yet see the white mycelium that we will, say, in a week from now. So yeah, you can find sclerotinia on the pods, you can find it on the leaves, you can find it on the stems, but when you look for sclerotinia, you're not looking for a hard stem canker, you're looking for a soft, mushy, wet lesion. And it will probably be pale white in color. And if you squeeze it with your finger, the fungus will have degraded all of the fibrous material. Well, it will degraded all of the sort of glue material holding the stem together, and all that's left are the fibers. So if you pinch it, there won't be any. That's why they lodge, is because the integrity of the stem has been compromised. And if you roll it between your hands, it'll just shred to pieces. So with blackleg, the stem tissue stays hard and woody, but it becomes brittle and it'll break off. Sclerotinia is soft, and if you rub it, the stem will shred, because all that's left is the fibrous material, and the other material of the stem's been digested by the fungus. The most problematic lesions caused by sclerotinia are the ones on the main stem, because what happens is that the stem loses its ability to translocate water and nutrients up and photosynthetes down. So it loses its ability to transport things through the stem. So basically everything above that lesion is dead. All the pods above that lesion fail to fill. So it's quite damaging as well. Like blackleg, it can be catastrophic when the disease pressure is high in a susceptible field. So clubroot. How would we scout for clubroot? Would we look on the pods? Okay, we'd want to look on the roots, because that's where the disease is going to show up. And what would we look for? Deformities, galls, tumors, big lumps or balls on the roots. There are some things that can cause unusual root symptoms, like balls or nodules, that aren't caused by the clubroot pathogen. So if you found a field that had a few plants that had some balls or weird swellings on the roots, what would we do? Shoot, shovel, and shut up. That's option one, I guess. Option two?
Call you guys.
Yeah, we'd want to call somebody who could give us a diagnosis to figure out what it is. And so if you want an answer, clubroot or not clubroot, there are laboratories that can do a molecular test that will confirm whether the pathogen is there or not. So if you see some unusual root, so in southern Alberta we don't really have clubroot at epidemic proportions like there are in some areas in central Alberta. But there are fields in southern Alberta, there are a few fields in southern Alberta that do have clubroot. The disease is spreading across the province. We hope that it'll be contained at some point and that we won't get it here in southern Alberta, but it is important for us to continue to look for it. The organism certainly can complete its life cycle and cause problems in our fields, and if it were to arrive here, it would be problematic for canola producers for sure. So if we scout for the disease and we find something that's suspect, we would send it to a laboratory for an analysis to see, is it clubroot or is it something else, Rob?
So probably you'd be looking in kind of maybe patches and field entry points in different places like that, no?
Yeah, so that's a good point. Rob says when you're scouting for this disease, there's places where you're more likely to find it than others. And we're more likely to find clubroot at the entrance to the field, or in low spots, or if you're driving by a field and you see a big patch that's dying or prematurely ripening, It could be sclerotinia, it could be blackleg, it could be clubroot. And the only way to know which one it is to get out and have a look. And the only way to know if it's clubroot is to pull up some roots and look. So those are the places where you'd be most likely to find it. When we do surveys, we do systematic transects through the field where we sample at various points that are a certain distance apart. But we also make sure we look in the areas that Rob just mentioned as sort of, in addition to the surveillance, the systematic surveillance, a scouting for the disease as well. And we're more likely to find it at the field entrance and in the lower spots or areas where we can see above ground symptoms. Now, we don't always see above ground symptoms when there are clubroot symptoms below the ground. And that's the reason we also do the systematic surveillance, is because sometimes you won't see any above ground symptoms, but the clubroot organism is still there and the populations are building to the point where then you will see the above ground symptoms. Did you have any other comments, Ron, on canola diseases?
I think with clubroot, a good time to actually scout for clubroot is after the crop's swathed. Because as Mike mentioned, it can be very difficult to walk through a standing crop. But with clubroot, you can walk out in the field, dig up the plants rather than pull them, because at that point the ground's probably dried out and the clubroot galls may be maturing and they'll break off if you just pull up the stems. But we quite often do a lot of our clubroot surveys post-harvest. You can go through the fields easily, you can dig the plants up, and you look for the remnant galls on the stems. So that's a disease where you can actually scout for it after harvest. Okay, any questions on canola diseases?
Can you guys maybe comment on the visual difference between alternaria spots on pods and cabbage seed pod weevil exit holes? Those are the exit holes?
Yeah, so the question here is differences between what you would see on alternaria black spot symptoms as opposed to cabbage seed pod weevil exit holes. And sometimes that's going to be difficult to determine the difference. And sometimes the cabbage seed pod weevil exit hole can be an entry point for a black spot. So sometimes you'd see both things happening. I guess that if you just see a hole and sort of some general just plant wounding symptoms around the hole, that's less likely to have an alternaria infection. It's when you really see the blackening symptom that's starting to spread out from the exit hole, that's when you'd be more suspicious of alternaria. Did you have anything to add to that, Ron?
I think with alternaria, it'd be very rare to see just one lesion on the plant. Usually there'll be multiple lesions on the leaves, stems, or pods. So, whereas cabbage seed pod weevil would have a limited number of exit holes, I'm thinking. So look at the whole plant and make your determination there. And if you had a question, you could get it into a specialist like ourselves. We could have a look at it under the microscope and help you sort it out.
So just after a really extensive survey yesterday, it was one field. I just noticed that there was quite a few lagus, and you could sure see, you just kind of noticed it on the pods because this was a field that was hilly and it was quite late, and there was quite a few lagus in the one patch that I was in, and so they might have been attracted into that area, but you can sort of see, sometimes you can kind of see something like kind of a blackening sort of thing from that damage as well.
Feeding injury.
Yeah.
Yeah, so sometimes insects can be a predisposing factor or entry point for diseases. So if there's no other, did you have a comment?
No,
Ron?
I think we'll just head down to the peas by the corn there.
So that's all we have. If there's no other questions on canola or cereals, we'll move down to the pea plots. So Greg from the Canola Council sends, the best thing that you can put into your field is your shadow. And so when you're scouting for diseases, most times you can't do that from the edge or from the road, but you have to walk into the field. So feel free to come on in. Now, because this is a research plot, we're not going to pull plants out and trample things, but feel free to come on in and have a look. Please come in. Come into the field. Because if you want to scout for diseases, you have to come in. Very good. Okay, so pea diseases. We're pushing up against a little bit of a time barrier here, so I'm going to just talk briefly about a couple of pea diseases and then spend some time talking about a survey and then leave the rest of the time open to you to ask whatever question is on your mind with respect to pulse diseases. So for peas, really the major disease problem we have in our pea fields in southern Alberta, at least until recently, has been Mycosphaerella blight or Ascochyta blight. So this is a complex of organisms, Ascochyta and Mycosphaerella, that cause a foliar or stem lesioning or blight of the plants. And it's not difficult to find it in these plots. And the reason is that we're testing for efficacy of some fungicides or timing of fungicides to control pea diseases. So if you come down and look at some of these pea stems and pea roots, you'll see this distinct lesioning that's caused by the Ascochyta blight complex, Ascochyta Mycosphaerella blight. Now, this disease, you can walk into just about any pea field in Alberta and find some level of Mycosphaerella or Ascochyta, but in some cases it's much worse, and that'll obviously have to do with the field history and conditions, the seed source, etc., etc. But we find it at low levels in just about every field, and it kind of hangs out low in the canopy, down low, and when the conditions are conducive for it to move up, it does. And it can move quite explosively because, as Ron mentioned before, for some of the cereal diseases, it's also polycyclic, and so it can produce numerous rounds of sporulation within the season. So it can move very quickly up the canopy. And you know, a low level of disease down low isn't going to cause a lot of serious economic damage, but it can compromise the integrity of the stem if the lesions down low on the stem get too big. And so it can predispose the crop to lodge, which then makes harvesting a challenge for pea growers. So it's a fine line you walk between, do I apply a fungicide to manage the disease, or is it not at an economic level yet with respect to reducing yield or making harvest time more difficult. But really the only option we have to control the disease when it becomes problematic in season is fungicides. We don't have any strong genetic resistance, and the disease can come in even if the seed is clean and had a seed treatment applied. The organism still can spread to new fields within the season. So fungicides are an important part of managing the disease. So that's all I'm going to say about Mycosphaerella, unless there's questions or comments about it. So Mike, is this the night spraying?
This is our night spraying fungicide study. So we've got 3 timings, just like we did with the herbicide trial that we have. So we've got a daytime spray date that we did 12 to 1 o'clock. We did nighttime from 12 till 1 in the morning, and then we did early morning 4 till 5 a.m. And we've got 3 different chemicals for the peas. We did 4 crops. We did wheat, barley, peas, and canola. So we've got 3 different chemicals for each and 3 different timings for each, which makes for a lot of research plots. So this one, does it say the treatments over there on that little yellow sign, or does it just say the dates? Just the dates. I think we have Preaxor. It's got them on each individual plot. So if you're interested in how they fare, you can have a look at the plots. We're mostly interested in, though, we're not comparing fungicides for these trials. We're just comparing the timings of day, so whether or not we're seeing better efficacy during the day or night or early in the morning.
So is there just, it's a one-time application?
It's been a one-time application, yeah, and maybe we should look at doing a blanket application if it looks like the disease is going to get out of hand. So I'll probably talk with Mike and Ron after about that.
Any other questions about Mycosphaerella?
Did you say that what's the difference between Ascochyta and visually?
Yeah, you won't be able to see the difference visually. The symptoms will be pretty much identical, and that complex of organisms essentially causes the condition. It can be either one or both of the organisms that cause the situation. Okay, so another disease in peas, it's not as common, but it can present itself in some fields, is sclerotinia. So white mold on peas can be a problem from time to time. Again, we don't have resistant cultivars. We don't have any pea cultivars. We don't have any resistance to white mold in any of our crops. And so fungicides are relied upon in the cases where sclerotinia would show up. So the last thing I wanted to talk about was root rots in peas, and this has become an increasing problem for Alberta pea growers. And as a result of the increased problem and kind of awareness of root rots in peas, and I think probably our wet springs of late have complicated the problems as well. There is a research proposal that was funded last year that involves surveillance, variety screening, and also looking for the impact of seed treatments to manage root rots in peas. And so this project is led by Dr. Shama Chatterton at Lethbridge, and we at Brooks, Dr. Ron Howard and I are also involved in the project at Brooks, and it involves provincial surveillance for the disease. We also collect and isolate the organisms that are responsible for the root rot so that we can identify what it is we're dealing with. And then in year 2 and 3 of the study, we'll really focus on management of the disease, either through varieties that maybe are a little more tolerant to root rots, seed treatments that can at least help for the first 10 days to 2 to 3 weeks, depending on conditions, to help give the plant a fighting chance to get established and started. And also potentially looking at other methods or ways to manage the disease. So this year, primarily what we've done is survey. We've surveyed probably 100 and some fields across the province. We've collected symptomatic roots from a number of those fields, and we'll be identifying the causal agents and then repeating that work next year, and also next year beginning on some of the seed treatment work. So the root rot issue is becoming extremely problematic, and especially it seems to be problematic for producers that have been growing peas for multiple years. And so it could be that there's sort of a buildup of inoculum in the soil, as there are some of the fusarium survival structures that can survive for significant periods of time in the soil. So it seems to be building, and obviously the environment will have a role to play in how severe the disease is as well. But root rots are really becoming an increasing problem for pea growers, and so we'll be looking for some solutions. Any questions about pea diseases? This is your chance to talk and ask and discuss pea diseases before I turn the mic over to Ron to talk about corn. No burning pea questions. Okay, that's good.
Okay. Of course, in Alberta, we also grow processing peas in the Tabor area, and they tend to be harder hit by these diseases than the field peas do, especially the root rot. So, whereas although this study Mike mentioned is focused on field peas, we hope some of the information might also be helpful to the processing pea growers. All right, there's a corn plot over behind us here, and it's very healthy, so we're not going to go into it and find anything there. But how many of you scout cornfields? A few people? Okay. So at this time of the year in corn, what you're going to see are mostly the foliar disease issues. There's a small oval leaf spot called focus leaf spot that occurs on corn. It's pale colored. You'll also see bacterial stalk rot, which is actually caused by the washing into the whorl of the corn plant. The irrigation water carries soft rotting bacteria down into the whorl, and you'll see a rotting of the stem. In fact, the plants will die, and if you slice that stem open with your knife, you'll see it's rotten pretty much right from the crown up through into the stem. That's bacterial soft rot caused by Erwinia. There's a new disease that we're seeing in corn called Goss's wilt. We'll talk about that in the tent. Corn smut, or common smut, is a smut disease of corn where the ears get replaced with these big, huge smut galls. It's maybe more common in sweet corn than in field corn, but you occasionally see it. I haven't seen any head smut for a number of years in corn, but that is a smut that attacks the tassels, the top of the corn plant, and haven't seen that for a few years, but it has been found in Alberta previously. But perhaps the most common disease that we would see in corn is stalk rot, Fusarium stalk rot. And that weakens the stalks so that the plants sometimes topple over and they may die above the lower stem area. The plant will die. But the easiest time to scout for it is after harvest, where you go in and pull up or dig up some stalks, split them longitudinally with a knife, and look inside for a pinkish discoloration of the Fusarium fungus. And it's the same fungi that cause stalk rot in corn are the ones that go to cereals and cause head blight. In fact, the Fusarium graminearum fungus reproduces very well on corn. It produces more spores on corn stalks than it will on cereal heads. And so corn and cereals in a rotation are not a good idea because of that. The corn really sets the cereal crop up for potential problems with Fusarium root rot and head blight. Corn is also, it gets on the cobs in both sweet corn and seed corn, and there you end up with the same mycotoxin issues with grain corn as you would see with infected cereal grain. So we have very high levels of Fusarium stalk rot in our field corn in southern Alberta. Some of the surveys we did in 2010 and 2011 showed virtually every field we surveyed had some, and sometimes levels in excess of 50% of the stalks infected. So it's certainly there as a problem in corn and something to watch for. Okay, any final questions on peas or corn before we board the wagon? We're going to head across the road to a barley plot. Okay, we're standing in front of another of our night spraying fungicide plots here. And this is a 2-row barley. You'll notice that within the plots the barley's maturing, and leaf symptoms and so on are not that conspicuous because the plants are almost dead ripe. But the alleyways in between the plots here were mowed earlier, and the plants were set back, and now they're flushing again. And we can see good symptoms of net blotch and spot blotch, which are major leaf diseases. The same ones that go to wheat are here on the barley. There's a third leaf disease on barley called scald, and it's unique to barley and rye, and it causes a leaf blotching. The lesions are sort of a pale color with a dark brown margin, whereas the other leaf spots, the net blotch and spot blotch here, much like you would see on wheat. They're brownish initially, often covered with are surrounded by a yellow lesion. And in barley a few years ago, they, doing more research on this disease, found that there's 2 biotypes of net blotch. There's a so-called spot form, which forms an elliptical to circular spot on the leaves. It looks a lot like spot blotch, which is caused by a different organism, and it stays as fairly small spots. The characteristic or typical net form of net blotch, which is the one that we normally consider as the primary type of net blotch, creates more expanding lesions that often net together and cause more extensive blighting on the leaves, and Mike's got a leaf he's holding in his hand there. So we can see the net blotch, spot blotch complex working on the leaves, and they will get up onto the heads. They'll cause bloom blotches. Their main effect is to remove photosynthetic area, and which reduces yield and quality. It's key that if you have high disease pressure from these diseases, you may need to apply fungicides to protect the flag leaf, the flag -1, and some of the other leaves in the canopy if you can as well. These diseases are residue borne, so they will persist on the straw and be there the following season. They liberate spores in the spring, which infect the seedlings, and the diseases will cycle several times during the growing season, that polycyclic cycle that Mike mentioned. Other diseases that we might look for in a barley crop at this time of year would be Fusarium head blight. Again, these heads are getting mature, we usually, when the heads are still a bit green, is when you can see the pale colored florets indicating fusarium infection. We would look for covered smut on barley. Again, the bunt balls replace the kernels, and you can crush those in your fingers, and they'll get, you'll see that the bunt spores on your finger, which have a fishy smell. If you smell them, they smell fishy. Ergot also attacks barley, and like we saw in the wheat plots there, if you hunted here, you may be able to find some ergot bodies as well. And if we dug up some of the roots, we may be able to see some common root rot on these plants as well. So those are the diseases that we would look for at this time of the year. And most of those carry over on the crop residue, so that's why back-to-back barley is not a good idea if you've had any of the diseases I mentioned as a problem this year. There are some fungicide options to use on barley, although I would say barley is sprayed much less frequently than wheat is. Particularly silage barley, it's taken off just as the diseases are gaining momentum. But if you're growing malting barley, where high quality seed is essential, then you may want to consider using a fungicide as an in-season application. That's about all I was going to say on the barley. We'll give you a chance to walk through the plots, but before we do that, are there any questions on barley diseases? We'll see fungal leaf stripe on barley, which is a relatively rare disease. We found I had a sample come into our lab this year. We'll show you that one. Barley yellow dwarf, which is a virus disease of barley, that we occasionally will see that in some years. It causes chlorosis or yellowing of the plants, and they're usually stunted. But other than that, those are the major issues that we see in barley here in southern Alberta. Okay, well, we'll take 5 minutes. We'll let you walk through the plots. Probably the alleyways are where you're going to find the most conspicuous symptoms. Feel free to pluck some of the leaves off, have a close look at them, and pull up some plants, check the roots out if you'd like. We don't obviously have any raging epidemics going on here. We've got a fairly good stand of barley. We're relying on natural infection for the fungicide trials that we're doing here. And this crop's getting very close to harvest. Any questions? If not, feel free to have a look in the plots. Okay, well, just let me thank all of you again for coming this morning, and we enjoyed showing you the plots and hope that you learned a few new things. As I mentioned in the introduction, we have samples of literature here that you're welcome to take with you and begin to build a library of information on crop diseases. So to wind things up this morning, we'd just like to talk about maybe some of the new or unusual diseases that we're seeing out in the field, that there weren't infected plants there to show you today. The first one I'll show you is this disease on corn. It's called Goss's wilt, and you may be able to see the pale streaks, the lesions on the leaves here. And this is caused by a bacterium called Clavibacter michiganensis, subspecies nebraskensis, and it was first described in Nebraska a number of years ago and has since spread through the Midwestern United States through the major corn growing areas. And it was found in Manitoba in 2009 and has become quite extensive there. We picked up a few fields in Alberta this year, and we're in the process of doing a survey to try and find out how many fields have the disease. It's a vascular wilt disease, meaning it clogs the vascular tissues of the plants. It's systemic in the plant, so it eventually can kill the plant. And when the first outbreaks happened, there were large areas of fields that were killed by the disease. But breeders have made good progress in developing resistant varieties, and now there's a number of resistant hybrids, and that's become the major means of managing the disease. It does survive in crop residues, so corn stalks and parts of cobs and that sort of thing could allow the bacteria to survive to the next crop. So if you're in a continuous cropping situation with corn, that would certainly play into the hands of the disease. It also is favored by mechanical injuries. So when you have a lot of sandblasting, hail, insect damage, things like that that predispose the plants, then the bacteria, which are splash dispersed with water, will enter those wounds and colonize. And once it gets inside the plant, it will cause a brown discoloration of the stem. We brought some fact sheets up here which you're welcome to look at and take a copy if you'd like. So Goss's wilt is a new disease. We're hoping it'll be a minor problem here, but we don't know until, of course, we have a closer look at it. We know it's capable of attacking silage corn, seed corn, and sweet corn, and the survey that we'll be doing in the next several weeks will hopefully determine whether it's in all of those kinds of corn or perhaps mostly in the silage corn. The disease, as I mentioned, has had a history of causing quite a bit of damage, but fortunate thing is we've got many years of experience in dealing with it elsewhere, and although it's very extensive in southern Manitoba now, they are bringing in and using the resistant hybrids and other management techniques as means of controlling it. It can also attack things like green foxtail and some of the grassy weeds, so it will survive on those. Those are potential reservoir hosts for it. But corn is the host of economic importance. So it's called Goss's wilt, Goss', Goss's wilt, and it's a bacterial disease. The Clavibacter genus has a number of important plant pathogens. Within that group, we have bacterial ringrot of potato, which we occasionally see here in Alberta. We have bacterial canker of tomato, which we have seen in our greenhouses here, and also bacterial wilt of alfalfa, which fortunately we don't see much of anymore, but at one time it was an important disease. So this particular genus of bacterial pathogen, it seems to be adapted here to the prairie conditions. Another sample, Mike's going to talk about the peas and beans here, but maybe the last one I'll look at. Down here at the end there's a wheat sample that has bacterial leaf stripe on it. It's a variety is Glen, which is a fusarium tolerant variety. And earlier this season we've had reports of long stripes in the leaves of the wheat, which didn't really match with tan spot or septoria. And when we brought these leaves into the labs and checked them, they were just full of bacteria. And this, the organism Xanthomonas that causes this disease is seed-borne, and so must have come in with contaminated seed. Then with the showery, rainy weather that we had, it multiplied and it splash dispersed around. So it has shown up in a few fields.
Ron, how would that compare to the green grass bug, where it kind of had the long...
The cereal leaf beetle?
Wasn't there a green grass bug causing damage as well? Like that? Or
Cereal leaf beetle causes that streaking. But what it does is it actually eats out the epidermis. So if you look carefully, you'll see that it's been eaten out. Whereas this, there's no removal of the epidermis. It remains intact. It just has long strips in it. So,
What's the difference between bacterial leaf stripe and wheat streak mosaic virus visually?
Visually?
Can you tell?
Okay, well, yes, you could tell that with this bacterial leaf stripe, it's a dark stripe with a chlorotic halo around it. And under the microscope, you can see droplets of bacterial ooze that have dried on the leaf surface. And with this disease, there's no stunting of the plant. And there's, other than the affected leaf tissue, there's no yellowing other than the tissue that's affected. It will get up onto the glooms and cause a gloom blotch, a darkening of the glooms. Now with wheat streak mosaic, there you'll usually see prominent stunting and chlorosis of the whole plant. You'll see rolling up of the leaves, and if you have a hand lens, you may be able to see the wheat curl mite, which vectors that virus. Also with this bacterial stripe, it occurs more or less through the field, whereas with wheat streak mosaic, it often is an edge effect where it's come in from an adjacent field. The mites can't fly, or they're moved, blown around by the wind. So there's usually an edge effect where you get a prominent chlorosis and dieback of the plants, and as you go into the field, the symptoms peter out. With bacterial leaf stripe, you'd see it more likely in areas like close to pivot points and that sort of thing where you're getting maybe some extra moisture.
So how's that vectoring around, or how do those bacteria kind of get dispersed evenly within a field like that?
Sure. Well, the fact that in this particular field it was quite evenly distributed suggests it came with the seed. And many of the bacterial diseases are seed-borne. And then it grew systemically in the plant and was splash dispersed with the wind and stuff and rain that we had. And I imagine, you know, some hail damage, had it been there, would have aggravated it as well. So bacterial diseases are often seed-borne, and they multiply and spread very quickly. You see that in wheat, you see that in beans with the bacterial blight on beans. Very quickly and rapidly dispersed by driving wind and rain especially.
So is that like 1 or 2 years out, it's going to finally get rid of that?
I don't think it's long lived in the soil, no. In fact, there are really no control measures for this disease other than using disease-free seed, and very few seed labs would test for this bacterial blight. They'd have to have special media or anti-sera to test for it. It is a problem, becoming an increasing problem in the States. It's there's some attention now being turned to looking at breeding for resistance. We don't know whether copper fungicides would control this as the ones that are used, for example, on bacterial blight of beans. But none are registered presently, so we don't really have that option. Okay, so that's what I wanted to say on those two diseases. We also had a sample of fungal leaf stripe on barley. Unfortunately, the sample isn't here this morning, but by the book Diseases of Field Crops in Canada, we put some leaves there with net blotch and some leaves with bacterial stripe, and that's with fungal leaf stripe. It's a fungal origin, of course, but it forms a darker colored lesion versus the yellowish lesion with the bacterial leaf stripe, which is what we have an example of there. And in the book Diseases of Field Crops in Canada, which by the way is an excellent reference, there's a good picture of the fungal leaf stripe of barley there, which you can have a look at. It again is a seed and residue-borne disease. Okay, if there's no other questions, Mike Harding's going to talk a little bit about some of the samples we have here of canola and beans.
Okay, thanks, Ron. On the topic of cereal diseases, I'll just point out that I did bring in a sample here that's got stripe rust on it. So I know around here probably everybody's already seen that one, but if you haven't, or you'd like to have a look at this sample, there's some stripe rust symptoms on wheat. Okay, so let's start with the pulse diseases. One thing we haven't talked about today is dry beans, but in southern Alberta we have quite a few acres of dry edible bean, and one of the major production constraints is white mold, caused by Sclerotinia sclerotiorum, the same organism that causes the stem rot in canola. And so you can see here the white fluffy mycelium. If you look closely, you can even see that the fungus has already started to produce sclerotia, the small black irregularly shaped structures that'll drop down into the soil and survive over winter and be the source of inoculum for the next time there's a susceptible host. So a nice example of white mold, which is the same organism that causes the stem rot in canola, that we weren't able to actually see today because we're still a little too early for that here. Okay, so the other pulse disease that I have here is root rot on pea. And so we talked about this out in the field, but this crop is obviously pretty far along. But if you look closely at the roots, you can see, excuse me, a discoloration. You can see a reddening in one of these. There's necrotic streaks, and there's also some red streaks down here. And you can see a constriction here at the stem, which is kind of right at the soil line. And so this is really characteristic of the Fusarium root rot that we believe is kind of the number one problem for root rot in peas. So there's some nice examples of the symptoms there. And then finally,
Just on these, like for the study that you're doing, when are you going out to gather samples on that? Is that close to harvest, or are you doing that survey run?
so the question is, when are we going out to look at and sample the pea fields? The first round of surveillance that we do is prior to flowering, or kind of right at flowering. And that's kind of a key time point for this disease to show up. So oftentimes if the roots are compromised by root rot, you don't necessarily always see above ground symptoms until there's some significant stress put on the plant, either because it gets dry or because it's triggered to go, expend a lot of energy for reproduction. And so at flowering is kind of a time that we thought would be a good time to start seeing some of the above ground symptoms. And so the first time that we do the surveillance is just kind of at flowering or just prior to. And then we do a second round of surveillance, which is when we collect the root samples for isolation, which is after flowering is finished but before harvest. And so we've actually just last week completed the last of our second round of surveillance. And in the second round of surveillance, we didn't go back to every field. We just went back to the ones that had the greater problem and collected a bunch of samples from those fields. So about 20 fields across the province were sampled and have the organism isolated. So actually 2 different sampling times, one just kind of for surveillance, and then the second one for the collecting of isolates for identification. So the last one I'll show you here is Sclerotinia on canola. And so we don't have any stem rot symptoms. And as I mentioned, that's really the economic, that's where the economic damage on canola really happens, is when the stem gets infected with Sclerotinia. Basically everything above that infection point is lost potentially. And so we don't have any stem lesions, but we do have some leaf lesions. And so if you look carefully on the leaves, you can actually see the remnants of those petals or blossoms that have fallen down, either stuck to or lodged on the leaves or at the leaf axils. And so around those remnants of petals, because they're mostly decayed now, you can see a lot of chlorosis or yellowing. And that's the fungus growing out off of it. So it uses the blossoms or petals as they're dying. It uses that as a food source to start to grow. And once it starts to grow on those petals, it gains enough momentum that it can then attack the green tissues of the plant. And so you can see that it's starting to attack the leaf tissues and causing them to die, and you can see the chlorosis. And so when you get blossoms lodged at the axil, where the leaf meets the stem, it'll actually simultaneously infect the leaf and the stem. And that's where the problems really get bad. So those are some examples of some of the diseases that we've talked about today that we weren't able to see out in the field. Does anybody have any questions about diseases on pulses or oilseeds? Okay, well, I'll turn the time over to you. Oh, question here.
I got one, Mike. Sclerotinia, will it infect, or could it infect a canola plant at the base of the stem without a petal?
So that's a good question. Can Sclerotinia attack the green tissue of the plant, say at the base of the stem, right at the soil line? So for example, let's say some blossoms fell down onto the ground and Sclerotinia started growing on those. Could it then grow into the stem at the base of the stem? Or could sclerotia germinate to just form hyphae and grow through the soil and directly attack the stem without having the blossoms for food? We don't really see a lot of evidence for that second of just mycelium. So sometimes sclerotia germinate to form spore-bearing structures, and then the spores land on the blossoms and start to colonize and then grow into the green tissue. And sometimes sclerotia germinate and just form mycelium, the thread-like filaments that fungi are characteristic for. And those filaments will grow through the soil looking for decaying material to colonize and grow on. We don't really see any evidence that Sclerotinia will directly attack canola stems as hyphae. So if we saw that, I mean, if that was the case, we would see a lot of sort of basal lesions without any evidence of decaying tissue around it, but still seeing a stem rot. We don't very often see that, very rarely. Most of the time what we see is either decaying blossoms as a sort of a starter food for the spores at the crown of the plant, or more commonly on the leaves or lodged at the axils. That's the most common place that we see Sclerotinia. So we don't really have good evidence that that the sclerotia will germinate into hyphae and just directly attack. Now that's not the case for some other plants. So Sclerotinia has a very broad host range. In addition to canola, as I mentioned, it causes white mold on beans. It also causes stalk rot, root rot, and head rot in sunflower. And in sunflowers, Sclerotinia does not need petals or blossoms to colonize the root tissue or I believe even the crown or the stem of the plant. It can just, those sclerotia can germinate into hyphae and just start attacking the sunflower plant right away. In beans, what we see is most of the time evidence that the fungus got started on petals or decaying material. But the fieldmen for Viterra that scout these fields say that in wide row beans where they cultivate, whenever the soil is pushed up against the bean stems during cultivation, they see a real increase in Sclerotinia at those locations. So pushing soil up against the stems indicates that it's either, in some cases, there can be some direct attack, but more likely the fungus gets started on a decaying leaf, decaying blossom in the soil, and once it builds some momentum, then attacks the bean stem. But piling the soil up against the stems sometimes will increase the level of white mold. So that's a good question. For canola, I would say the answer is probably no, unless Ron's going to have more to add there. For canola, we don't see evidence of direct attack from hyphae, but for some other crops like sunflower, we do see that.
You mentioned with canola and beans, if one infected plant topples over onto another, you will see the bridging happening that way. So sort of plant to plant spread can happen with Sclerotinia that way. Okay, any other questions on the pulses or canola of Mike? Any questions on cereals or corn? Okay, let me just end off then with a few concluding remarks. So in the introduction, we talked about the importance of scouting your crops at harvest to look for problems, to determine where they're the worst in the fields, to determine the identity of these problems. And so what do we do with that information once we have it? Is it something we just file away, or do we actually make some practical use of it? Well, I'll give you 3 practical ways that you can use that information if you have time to collect it. Number 1 is just to improve your diagnostic abilities with crop diseases so that you'll learn to recognize things, the predominant diseases that are occurring on your crop. You can convey that to your children if they're helping you farm, or perhaps to hired help and so on. Or if you're hiring agronomists, to give them a bit of a background of what you know has been in your field. So it's having that building that base of knowledge and experience. Diagnosis improves with experience. That's the bottom line there. The second way you can use that information may be to help you manage the infected crop residues, the grain, the straw, and so on, that you're harvesting from those fields. So for example, if you had a field with Goss's wilt here, and you harvested it for processing corn, let's say, and the stalks are still out there, would you want to go in there with tillage machinery, potentially contaminate it with those stalks, and then go to several other fields on your farm, dragging that material with you? Or if you were using a chopper, say harvesting silage, going through a field with Goss's wilt, knowing the bacteria would get on the blades and so on, of just going on from field to field to field with that same piece of equipment without decontaminating it perhaps after harvesting a contaminated field. Well, there's things like that, or if you save your own seed, you'd want to know if there's some particular diseases, what the risk is of those diseases being transferred into the next crop. So you'd want to have some seed testing done. So it helps you, I think, with managing the seed and residues from those infected crops. And the third thing it'll help you do is to refine and improve your management strategies. If you've used fungicides that don't appear to be working, if you're growing varieties that appear to be ultra susceptible, or other practices, perhaps you're irrigating too much or too little, that seem to be predisposing the crop to disease issues, now's the time to assess those, or at least through the winter, and make whatever changes may be necessary for the coming year. So you are planning ahead, in a sense, by gathering information now that will help you to either validate the effectiveness of those measures or to pinpoint some problems which you can correct. So I'd just like to encourage you to, when you go home, make sure you have a good look at your fields for a lot of the diseases that we were talking about today or others, and build an inventory of what's there and use that information to your best advantage. Yes?
I have a field of spring wheats that I sprayed a Fusarium product on at flowering.
Yes.
On the wheat, and I did have some cereal leaf beetle damage on the leaves a little bit.
Yeah.
But it almost looked like, say, 7 days after spraying, it looked like my flag leaves looked worse off than they did when I sprayed it, which I wasn't quite expecting.
Right.
I didn't see that on my other fields. It was just the first field actually. Could that be something that's actually not a disease? That's just something caused by how the type of chemical I use? Or?
Well, yeah. I mean, sometimes some of these products have solvents and so on that may cause phytotoxicity on the leaves under certain conditions. That's true. And it could be as well that maybe there was a disease issue there that was at the early stages, when you were spraying. It wouldn't be controlled with the fungicide, and it's now like the bacterial leaf stripe, something like that. That just may have taken the opportunity to multiply and start.
It looked like a tan spot.
Did it look like?
Yeah.
There is a range of so-called physiological leaf spots that look exactly like tan spot, and they're often caused or triggered by certain environmental conditions, and you may have had some physiological leaf spot there. There's a couple of good fact sheets on physiological leaf spot. If you want to get a hold of me, I'll send those to you. It talks about the environmental conditions that favor physiological leaf spots, and they do look exactly like tan spot. They're all really hard to tell apart. Yeah. Okay, any other questions? Any other crops that you're growing that we haven't talked about today? Any questions on those?
I could just
Yes,
just
Rob.
flax, because there's a fair
Yep.
little bit of flax up there.
Yep.
So just again driving along, I see some of the flax. One flax field that was sprayed, it looked really good, and then the next one, you didn't see any tracks. I don't know if it was aerial sprayed, but
Right.
kind of walked out in a bit of a patch there, and it looked like, is that Pasmo? I don't know.
Well, we don't have a history of a lot of the flax diseases that you'd see in southern Manitoba, for example. And Pasmo, which is probably the major foliar disease of flax, it's a fungal disease, certainly could occur here. And I've seen over the years a few samples I thought had Pasmo, but I don't regard it as a problem. The things that we've seen this year on flax we saw some Rhizoctonia seedling blight at the early stages, causing constrictions on the stems, and they were toppling over. Sclerotinia will attack flax, and botrytis or gray mold will attack flax as well. We did see some flax plants with leaves that were dying. Then I did see some fusarium on the stem, and there is a fusarium wilt that attacks flax. it could have been that, but we really didn't have the time to pursue it. I've not seen a bona fide case of pasmo this year, to be honest with you. But, you know, it could be out there.
And hearing about pasmo, but I wondered if it's around, so that's good to know.
Yeah. So we've escaped the aster yellows bullet this year. That can affect flax, of course. Flax rust comes in very late. I don't think we'll ever see that as an issue here. But
Is there any way to control the seedling blights in flax?
it's seed treatments are about it, and seed bed preparation. That's about it. Yeah. You want to get the seedlings
Liquid
out.
treatments on flax don't work very well.
No, flax is a challenging crop. That's right. Yeah.
How about mustard? Is it similar diseases to canola?
Mustard does have similar diseases to canola, but if canola's up here for diseases, mustard's down here. It's actually usually a very healthy crop. You once in a while will see the seedling diseases and root rots in mustard, occasionally sclerotinia. Mustards are very susceptible to clubroot, but I've never seen it in, you know, a bona fide case in commercial mustard fields here. So it's actually a pretty good option, and mainly I think because we grow it under dryland conditions for the most part. If you were to irrigate the mustard, I think you'd see some of the same diseases that we see on canola. But mustard seems to have a better resistance package than canola. Yeah. Okay, any other questions? No? Okay, I'll turn it over to Jamie to close things off here.
Sure. Just first of all, thanks for everybody for coming. We appreciate the turnout. And how about we give a hand to Ron and Mike? And just wanted to give a quick thanks to our sponsors, because obviously without them we can't do events like this. So hopefully you guys learned something, and if you see one of our sponsors along the way, ACPC guys, Alberta Pulse Growers, Winnowee guys, all these people are helping put this stuff together and helping us learn. So if you see them, just give them a thanks, and we appreciate it. Any questions, feel free to contact us and we can pass you information.
Have a great harvest.